So good afternoon, everyone. My name is Anna, and on behalf of the team PPK, I'm here today to present to you Sensorium, tackling neurodegenerative disorders by improving protein aggregation detection. Neurodegenerative disorders are a very heterogeneous group of diseases where we can find Parkinson's disease, ALS, dementia, and even Alzheimer's. Take this one as an example. First, you start by forgetting your keys. Some months afterwards, you may not even remember who your loved ones are. And by the end of your life, you may be completely oblivious of who you are. And these extremely debilitating diseases already affect more than 100 million people worldwide. And this number is expected to increase since currently there is still no effective cure. And today, we are here to present a solution that will help pharmaceutical industries to find the best drug to treat neurodegeneration. So as I just told you, these are very heterogeneous disorders, but they all share a common feature, which is the accumulation of protein aggregates within cells. And what does this mean in the context of neurodegeneration? In a healthy cell, you always have cells producing healthy proteins that are essential for cellular functions. In fact, proteins are known as the building blocks of life, and without a protein, there is no function. In a disease cell, in a neurodegenerative disease cell, something goes terribly wrong with this process, and proteins become aberrant, and they accumulate in the form of protein aggregates inside the cells. And this is extremely deleterious for cells and extremely toxic, leading to cell death that ultimately affects tissue functions that is then responsible for the debilitation that is often observed in the patients from these disorders. And the pharmaceutical industry is well aware of this, and they know that protein aggregation is the best target to try to cure neurodegeneration. But until today, there is not a single drug in the market that can reverse this phenomenon. And this happens because really at the early stages of the drug development pipeline, even in the preclinical stages, there are currently no effective cellular models of protein aggregation that allow drug screenings to test drugs that can indeed reverse protein aggregation. But this is about to be a problem from the past, because we have developed a technology that can sense it, screen it, and solve it. And what is exactly our technology? We have genetically modified cells with a fluorescent sensor that detects protein aggregation. In other words, what we have is a molecule that is bound to a fluorescent dye that can recognize any kind of protein aggregate that occurs in cells. So if you remember the cell that I just shown you with the protein aggregates, if we have that same cell genetically modified with this fluorescent sensor, we will be able to observe under a fluorescence microscope the formation of those aggregates, as you can see here. Now imagine that you have a drug and you want to test its efficacy in reversing this protein aggregation state. In that case, you add the drug to the cells and wait to see what happens. By the end of the treatment, if you have even a reduction in size or a reduction in the number of protein aggregates, or in an ideal situation, a complete reversal of the protein aggregates, this means that this drug, this molecule, is a good molecule to test in the subsequent steps of the drug development. And indeed, our collaborators say that this is a great tool to have in cells to finally be able to perform drug screenings to find the exact drugs that can indeed reverse protein aggregation. And if we look at the drug discovery pipeline that is commonly used by the pharmaceutical industries, what you see here is that there are many stages until we get to only one drug. And by applying our technology at the early stages here, at the compound ID stage, we can allow the pharmaceutical industry to decrease the number of false positives that come out from those screenings, while increasing the likelihood of finding a true drug that can indeed reverse or at least modulate protein aggregation. And to deliver this to our potential customers that are pharmaceutical industries, research institutes, and CROs that are interested in testing the efficiency of their compounds in the reversal of protein aggregation, we propose to have a service, a drug screening service, where our customers just deliver their compounds to us, and we perform all the testings until we deliver a report where we identify the best hits. So we are already testing this business model with a collaborator of ours, ExtremoChem. So we are testing the compounds that they sent to us, And by only using our technology, we were already able to identify a promising compound, as this one that you can see here, that can reduce around 40% of protein aggregation. And this was using only our technology. So we are the only ones that can deliver integrated results, meaning that we can perform lifestyle imaging, allowing to monitor all the process, while we are detecting multi-protein aggregation, whereas our competitors, Proteostat and Inoprot, can only deliver this separately. Moreover, we are better than our competitors because we can deliver these results with high accuracy and at high result speed, and we can produce closer disease models. So the drug screening services on their own are already a market. They represent a market of around $600 million, and this is expected to increase up to $1 billion by 2024. So to enter this market that we envision we can be in by mid-2020, we will need an investment of 2 million euros. And these 2 million euros will allow us to finish our proof of concept, go through process automation, and setting up our facilities. After entering the market, we believe we can reach the break-even 1.5 years afterwards, and we can reach a net profit of more than 35 million euros in 2024. And we are also planning already a new round of R&D from the stage we enter the market on. So let me just finish by presenting to you our team. We are a team composed of 3 researchers that work together for more than 5 years now, and an MBA student that cumulative have more than 30 years of research experience. We were highly advised throughout this process by our 2 mentors, Carlos and Rita, and we are very willing to bring this technology to the market. And before I go, let me just tell you something. The next time you forget your keys, don't worry too much, because we are working on it. Thank you.